last time when we were in class, I went through the outline of what is expected. So I think I'll go straight to the this slide, this one, which is talking about the difference between the glial cells and the neurons. Yeah, because one of the things we are supposed to look at is the structure and the types of neurons, and then their function, where they are located, and how they work. So I'll go straight to cells of the nervous system. So we have two main types of cells. We have yes. So we have the glial cells and we have the neurons. Now, if we take the whole of the nervous system, it is generally divided into two portions. The two major regions being the CNS, central nervous system, which is made up of the brain and spinal cord, and the peripheral nervous system. And those are the nerves that originate from the CNS and they go to the peripheral parts of your body. Now, if you look at the two types of cells, neuron and glial cells, the glial cells are the ones that give the supporting function to the neurons or to the nervous system. How do they support? One, they give the framework. They give actually, if you look at, for instance, if I talk about the axons, I told you that the diameter of the axon is very, very small. Yet the function of the axon is crucial for your survival. So this has to be protected. And the cells that protect it are the glial cells. They present a cushion, and these glial cells also help in providing nutrients to the neurons. So they give the mechanical support. They give, I would say, they protect even from harmful effects. So they give quite a number of support, both structurally and functionally. Now, on the other hand, the neuron is the functional unit of the nerve. It's the one that is able to transmit electrical impulses. What do I mean by that? It can generate and conduct electrical currents. So that function is the main way that your body is able to communicate. So it must be able to generate, and number two, once it generates, it must be able to conduct. And usually after conducting, it has to pass the signal to the adjacent cells. So if you look at those two types of cells, both the glial and the neurons, you find that you have multitudes of these cells in your body, where if you look at just the CNS alone, we are talking of about 100 billion of the neurons, the conducting cells. Now, if you take 100 billion, just multiply that by about 50 times, and now you get the number of glial cells. So you find that the glial cells are more numerous. They may be the ones which are not doing the function of electrical signaling, but they are the ones which are more numerous so that they can support the functional cells. Now, for that reason, we say the neurons are the excitable and the glial cells are the non-excitable. Now, let us first focus on the glial cells. Then we'll be able to go and look at the structure of the neurons. The glial cells can be divided into two categories. There are those that are found within the CNS, and then those that are found within the PNS. Now, I'll start with the PNS because you are more familiar with it. In the peripheral nervous system, we have neurons that have axons, and those axons may be myelinated. And from our previous course, we saw that the myelin sheath is made up of Schwann cells. So Schwann cells are the first glial cells that you have encountered. They form the myelin sheath in the peripheral nervous system. And we are going to see their importance, especially when it comes to conduction of electrical impulses. For number one, they give the support to the axons, but number two, they also help in speeding up. They are not the ones that generate the electrical currents, but their presence will speed up the rate at which the impulses move along the axon. The second, you find that I have mentioned the Schwann cells usually wrap around the axon. And you find that they have, we'll discuss the myelin sheath and how it is formed. But I always tell people to use the imagination and think of something like a balloon. If you have a balloon and you put a golf ball inside the balloon, and this balloon is not inflated, so you tie at that point and you have a golf ball inside and you don't inflate it, then you can take something like a rod. Just take a big stick or a rod and you place it on one end of this balloon, then you wrap around it. So now what you'll be doing is you wrap around so that this part is on this end. Actually, I should have put it at the other end. Let's see. So if I have the stick here at this portion, so now I start wrapping the balloon around this rod. So I'll throw the balloon and wrap it and wrap it and wrap it. So what I will end up is having a spiral shape here of these going under and wrapping and coming back under and wrapping and coming back under and wrapping. Now the interesting thing is because you have this golf ball, as you wrap around, the golf ball will be thrown to the surface. It cannot be stuck under there. So every time you wrap, this ball is on the surface. So at the end of the day, what you're going to have is you will have the myelin sheath wrapping around. So you have the part where the sheath is. And now because of this bulge here, of the, I would say in this case, in your case, it is the golf ball. But in our case, we'll be talking of the nucleus. So the golf ball is acting like the nucleus. You find that the Schwann cell will always have one part that is bulged up, making up the position where the nucleus is. But what is underneath is the tight wrapping of the two membranes. So remember, if you have a balloon, you have the rubber on top of the golf ball and you have the rubber part that is beneath the golf ball. Those rubber parts are representing the cell membrane of the Schwann cell. So as you wrap around, you are going to squeeze out the cytoplasm, and the cytoplasm together with the nucleus will be located on this end. So what you have tightly wrapping in the inner section are the two membranes opposed to one another without a cytoplasm in the middle. So we just have a layer of phospholipid bilayer, another one and another one and another one. It is only towards the surface that you have some cytoplasm, and you're also going to have the nucleus located there. So for that reason, we say that the myelin sheath is actually made up of the components of the cell membrane of a Schwann cell. Now, we have discussed the Schwann cell wrapping around the axon. So what happens to the cell body? If you remember, a neuron will usually have a nucleus and a cell body, probably with dendrites, extensions, and then it will have an axon, which is usually the long cytoplasmic extension. So around this part, we have already said you have Schwann cells. And these Schwann cells are the ones which make up the myelin sheath. So now, what happens to where there is no Schwann cell? And you find this is at the cell body. The cell bodies do not have the Schwann cells. So this will make this area very vulnerable. Now, in cases where we have aggregation of what are they called, the cell bodies, you may find that in between we have some cells that come and oppose themselves on the surface of the cell body. These cells will give protection to the cell body, and they're the ones which are known as the satellite cells. And most of the time where you're going to find them is usually at an aggregation of cell bodies outside the CNS. So if you call, do you remember what you call an aggregation of cell bodies that are located outside the CNS? Who can remember? When we have cell bodies, but they are all aggregated at one point, but the location is outside the CNS. Yes, Lutendo. Unmute yourself, please. So what did you call them?
Ganglia.
Pardon? I can hardly hear. Lutendo. Priska, I see you have also unmuted yourself.
Ganglion.
It is a ganglion. So you'll usually find them around the ganglion. That's where you find all these cell bodies. And then you'll find a lot of these satellite cells, and they oppose themselves, they flatten themselves on the cell bodies. Now, in addition to protecting the cell bodies, they also act like macrophages, so that if there is anything, any debris that needs to be cleared around the cell, remember that neurons require a very, very clean environment, because if we have debris, you might change the pH. If you alter pH, you open channels. The minute you open channels, you start initiating action potential. So you don't want that to happen. So you have to keep these areas around the neurons very clean. And one of the functions of the satellite cells is to act like macrophages. They clean up areas around the cell body. Now you find that when we have neurons with axons, the axon arrangement is usually they have connective tissues in between the axons. So if we have quite a number of axons running parallel, and then they are organized into a nerve, in between the axons we have connective tissue. And together with the connective tissue, we have blood vessels. Now, one of the things that we find rich in these areas are macrophages, the real macrophage cells that we find in between tissue, and their work is to phagocytose or to engulf unwanted material. So within the nerves, we have quite numerous macrophages, but around the cell bodies, usually the satellite cells are the ones which act like the macrophages. So those are the two major types of glial cells that we find in the peripheral nervous system. Now, if we go to the central nervous system, we're going to find 4 types of cells. And these are the microglia, astrocytes, oligodendrocytes, and epidermal cells. And we'll go through all of them individually. So let's start with the equivalent of the Schwann cells. I told you that if you don't find Schwann cells, in the CNS, because they are only found in the PNS. There must be an equivalent cell that acts like that. So there is a cell that is going to provide the myelin sheath, and this cell is known as the oligodendrocyte. So if you want to remember that name, you have to split it into 3. Oligodendrocyte. A cyte is a cell. Dendrocyte. So it has dendrites, but then it is an oligo, so it has all these cytoplasmic extensions which do not have a given form of shape. So now if I were to look at this oligodendrocyte, you can see this one here. Now, if you see, it has multiple cell extensions as compared to the Schwann cell. So if you are, I'll take you slowly here. This area is within the brain. This diagram, schematic diagram you've been given here, represents the brain tissue. And among the tissue, we have different cell types. So I'm focusing now only on the oligodendrocytes. So if I was looking at a neuron in the peripheral nervous system, I am going to have the axon, and within that axon, I will have a single Schwann cell wrapping around one area. Then next to it, I will have another single Schwann cell wrapping around that area. So you find that one axon in the PNS is myelinated, or the number of cells that form the Schwann cell, that form the myelin sheath, are numerous Schwann cells, and they all lie in series, and then they form a continuous myelin sheath. This is in the PNS, okay? So in the PNS, multiple Schwann cells form a myelin sheath along a single axon. Let's go to the brain tissue. One axon, you can see this, what I'm showing you here, is the cell body of one neuron. It has an axon extending this way. Now you will find the first part forming the myelin sheath may be a cytoplasmic extension of one oligodendrocyte. The next part is actually coming from a different oligodendrocyte. The next part may come from the same oligodendrocyte or from a different one. So you'll find that we'll have all these not Schwann cells, myelin cells, myelin forming cells, but they are originating from an oligodendrocyte, but not a single one. There will be multiple oligodendrocytes aligning themselves to myelinate one axon. Similarly, a single oligodendrocyte can myelinate several neurons. So it may go to myelinate this neuron, then it forms an extension to myelinate another neuron, and another extension may go to the same neuron, or it may go to a different neuron. So one of the differences between the Schwann cell and the oligodendrocyte is that one oligodendrocyte can myelinate several axons. They can also myelinate the same axon but at different places, so that it will form a cytoplasmic extension, myelinate this part, form another extension, myelinate another part of the same axon, which is unlike what I have described in the Schwann cell. So I hope you are still following. Now we move from the equivalent of Schwann cell and we go to the equivalent of macrophages. So we say the satellite cells act like macrophages. So we also have cells in the brain that act like macrophages, and these are called microglia. Now, the reason they are called micro is because they are the tiniest. They are the tiniest of these glial cells. You can see them here. If you look at the volume they occupy or the space they occupy, they usually just have the nucleus and a very thin layer of cytoplasm. But what you have is very many extensions of the cytoplasm, and they are extending in a spider-like or web-like structure, going to all directions, and they have they're filling the environment. Number two is that they are not stationed in one place. These microglia, they are always moving around. So they are crawling around like spiders with these cytoplasmic extensions acting like pillars, trying to fill the environment. So what happens, because they are so tiny, how are they going to engulf? Remember, macrophages usually engulf unwanted material. So how does the microglia engulf, and yet it is so tiny? So now I will tell you how the microglia works. As it crawls along and it comes and encounters a debris somewhere, so these fillers will feel that that item is not supposed to be there. It will cause activation of the microglia, and one of the first steps that the microglia will do, it will withdraw these cytoplasmic extensions. Now, once it withdraws it, remember, this is a law, shall we say, it is one of the laws of matter. You find that matter can neither be created nor destroyed. It can only be shifted somewhere. So as you form cytoplasmic extension, you usually deprive the cell body of the cytoplasm because it is extending in these extensions. So now what happens when we withdraw extension, the microglia starts to swell up and the extensions are minimal. So you find only a small extensions, which can actually be flattened so that you end up having more of a microglia with very tiny extensions like that. Now, when you have an activated microglia in this format, then it can engulf the unwanted debris. Once it engulfs, it has the ability to destroy it, and after destroying, it becomes inactivated and it starts to throw out extension. And as it does so, the volume of the cytoplasm becomes smaller and smaller, and it retains its small shape, and it is able to start crawling again. So we say that it is called a microglia because it is very tiny as compared to the others. So this is the only very small cell. All the others are large cells, and for that reason, they are called macroglia. So we only have one microglia, but all these other cells are macroglia. So among them, we have already discussed the oligodendrocytes. So which are the other two macroglia? These will be the astrocytes and the ependymal cells. So I will discuss the astrocytes first. But before I go to the astrocytes, I want to know if anybody has a question so far. No questions?
Madam,
Yes,
Madam,
Condi?
okay. When you were talking about the how like microglia engulfs the unwanted materials, I
Yeah.
didn't understand. So kindly may you repeat?
The activation. We talk of activation of the microglia, and when it is activated, it will withdraw or it will pull back the cytoplasmic extensions. So I'll come back to you, Nema. If you have a cell like that, and then you pull out one part, and together with that one elongated part, you pull some cytoplasm. What you're doing is removing a small volume from the cell. If I make another extension here, and I pull from the cytoplasm here, the volume becomes smaller. Now you can imagine when I have multiple extensions in all directions, you find that you'll hardly have space in the cell body, so that you'll only have the nucleus and cytoplasmic extensions and a very, very thin layer of cytoplasm here. Now, when any of these extensions encounter something that is unwanted, then it will send information to the nucleus, and it will become, not to the nucleus, to the cell body. And what it will do, it will start pulling back these extensions. And as it pulls back, it brings about cytoplasm with it. All these will bring cytoplasm, and it will retain the spherical shape that we normally find of a cell. So now it will become a very large cell with minimal cytoplasmic extension. And now when it is in this state, it is ready to engulf the material. Okay?
Yes.
All right. So now, Nema.
Madam, it's clear. The same question.
Oh, you had the same question.
Yes.
So you are all good. I move on. So remember so far, in the CNS, always remember 4 different types of cells in the CNS. In the peripheral nervous system, there are 2 different types of glial cells. So now we have discussed those of the PNS. Now we are discussing of the CNS, and in the CNS, I have already discussed 2. So that leaves me with 2 other supporting cells, and that is the astrocyte and the ependymal cells. So I will focus on the astrocytes first. Now the astrocytes will also throw cytoplasmic extensions in a similar manner with the oligodendrocytes. The only thing that it will not do, their cytoplasmic extensions are not going to form a wrap around any axon. They will not wrap around the axon. What they do is, when you find this extension, it comes to the surface of a neuron, or it comes to the surface of a blood vessel. It will flatten out. And when it flattens out, it will form a big area that is covered by that cytoplasm from the astrocyte. The next astrocyte will cover another area, and the next astrocyte will cover another area. So assuming that this is a tube-like structure, what you will end up having is, if I have this tube here, let me just draw a tube there. And I assume that I have a cytoplasmic extension, and I come and flatten it out on that part of the tube. Then I have another cytoplasmic extension from another astrocyte, and I flatten it against this part. And I have another one coming from here, and I flatten the cytoplasm around this part. I bring another astrocyte extension, and I flatten it against this part. So you'll find that this vessel is going to be wrapped around, totally engulfed by these flat, like I would say, like podocytes, like flat feet. And they are all coming and flattening around the tube. Now, the interesting thing is that they form very tight junctions between these cells, so that they cannot allow anything to leak between them. So when they form this, it means that this vessel has been completely covered by multiple podocytes or extensions which have flattened on them. And because nothing can leak out, if you have blood passing through the lumen of this blood vessel, and it leaks out, say it is a capillary, and it leaks out in between the spaces of the epithelial cells of the capillary, you find that whatever leaks out cannot be able to pass to the brain environment because the brain has been separated from the blood by these podocytes, these extensions. So they form essentially a blanket around the vessels, and that blanket is what we call the blood-brain barrier. How many are lost? Blood-brain barrier, the formation of the blood-brain barrier. So you are all with me?
Yes, ma'am.
Okay, Selina's hand was up. I don't know whether she was lost or she's with me. Selina,
Please,
you had a question? Pardon?
I was wondering if you could go over it.
Okay, so if I have an astrocyte, I will erase this diagram. Say I have an astrocyte here. This is the nucleus of the astrocyte. So it has cytoplasm, and then it throws an extension here. And it throws another extension there, and it throws another extension there. So this astrocyte, when this extension goes and comes across a vessel like this one, that extension at the terminal will form a flattened area. And this flattened area will lay against the blood vessel. It will cover it, but it cannot wrap around it. It will just cover a given area, a small area of the blood. This other extension that comes here covers the next adjacent area. We have another adjacent area that is covered maybe by a different astrocyte here. So this one has some cytoplasmic extensions here. And this one covers also this area. So you can see, like in this case, I have drawn two astrocytes with a nucleus, one here and the other one here. So probably I have a third astrocyte here. And this one will cover this area. So you can see we have cytoplasmic extensions coming from different astrocytes. When they come and they lay against the blood vessel, they just flatten. That it forms like a bulb, a terminal bulb, but the bulb is usually round. But now imagine you flatten the bulb and it flattens on that cell membrane, on this vessel. Now, where it flattens at the edge of one astrocyte, you'll find that it will come in contact with the edge of the other adjacent astrocyte extension. Then this comes in contact with the other one. Now, what will happen is they will form tight junctions between them. So we have this astrocyte A with its extension here, forming tight junction with the adjacent cytoplasmic extension coming from astrocyte B. And when they do that, it means nothing can pass here. Now imagine this is the arrangement all around. So the whole of the periphery of this vessel has been surrounded by these podocytes here. So it forms all these tight junctions. Now it means that nothing can leak from the lumen here. So if we have blood, this is a blood vessel, and blood is passing through the vessel. If it leaks from the blood vessel and it comes outside, it cannot go into the brain space because the astrocytes are residing in the brain. And that space of the brain requires a tight regulation we need a specific environment. We do not want debris coming from the blood. They will change the pH of that environment. If we have any microbes coming, you are going to destroy these parts of the brain. You can easily infect the cells of the brain. So you need to keep the brain compartment, this area where these cells are, you have to tightly keep it away from the blood. Blood comes with toxins. It comes with so many things. So now the astrocytes are forming a barrier between the blood vessel and the brain compartment. For that reason, they are called the blood-brain barrier. They are the ones that are forming the blood-brain barrier. Now, if we need anything to go into the brain compartment, if we need anything to go into the brain compartment, because these cells have to be nourished, they have to be provided with stuff, then we will go to a fourth type of cells which I'm going to explain, and that is the one that decides what should pass into the brain. They take stuff away from the blood, they manufacture their own fluid, and they decide this is what can be available to the brain. And once they avail it, they pass it on to this other side into the brain tissue. So now the brain cells will be nourished by stuff that has been selected by these other type of cells, which are the ependymal cells. So for now, we just need to know that the brain compartment is separated from the blood compartment by the astrocytes. Are you okay now with astrocytes?
Yes, ma'am. Thank you.
Okay, welcome. So now, Rutendo, your hand is up.
Can you hear me?
Yes.
Okay. Ma'am, I understood about the blood barrier part that they prevent leakages from the blood vessels inside the brain, right?
Yes, yes.
From the diagram, I would like to understand how the astrocytes connect to the neurons.
Now, remember that these cells do not have a brain in them. All they do is throw out cytoplasmic extensions. And when they come across this flattened cell membrane, they lay against it. They just flatten on it. So it could be a blood vessel. It could be any of these. So all it is doing, it is forming a barrier. But the most important one that we know is that its main function, it will separate the brain compartment from the blood compartment. But for these other parts, it may form a protective function. Remember that we said satellite cells will form a protective function of the cell bodies in the peripheral nervous system. Can you see that the cell bodies here are naked? So the astrocytes are also giving that protective function on the cell bodies within the brain. So that would be the function that is carried out when they are forming these flattened or podocytes, because the way they are, you can see they are flattened here. This is the same way the satellite cell will come and flatten itself against a cell body within the ganglion in the PNS. So you find a satellite cell will come and flatten here, another one will come and flatten here, and they protect the cell body within the PNS. But in this case, this function is being carried out by the astrocytes. Okay, Nema? Your hand is up, Nema?
Yes, I just wanted to understand the other significance of the astrocyte other than being a barrier.
Now, the astrocytes mainly their work is to form the blood-brain barrier. That is the major function. But you can see that I have also said they have that protective function. Now, you can see that we have said that we have two types of astrocytes. We have those fibrous type and then we have protoplasmic type. Now, if you see the fibrous ones, they usually form those we call the intermediate filaments. These filaments are they sort of like part of the cytoskeleton. So with the cytoskeleton, you can have the microfilaments, you can have the intermediate filaments, and then you can have the microtubules. So these intermediate filaments are giving support, structural support, and you can see if they're within the white matter. White matter, oh dear, white matter is arising due to the presence of the myelin sheath. Now I'm jumping the gun because when I start discussing the myelin sheath, you see that it is mainly made up of phospholipids. Lipids appear white or yellowish in color. So if you cut a section through the spinal cord or the brain, you're going to cut through some areas that are mainly made up of the myelinated neurons. If they are myelinated, then those areas appear white. Now, because they are myelinated neurons, they need more of the structural support. And that's why they have the filaments, or they are rich in the intermediate filaments, because they need that structural support. On the other hand, you find that we can have the protoplasmic astrocytes. These are the ones which you'll find around the gray matter. The gray matter is devoid of the myelin sheaths. This gray matter usually has the cell bodies and the non-myelinated neurons or axons. For that reason, you find that if you have a lot of cell bodies, this is where a lot of synthesis occurs. And with synthesis, there is a lot of granular matter. So the astrocytes around the gray matter are usually granular. Okay? So this just describes the two types of astrocytes. Those that are found within the white matter, they are more filamentous, and those that are found within the gray matter, those ones are more granular. So I don't know whether that answers your question. I've added some information, but also tried to capture your question in it. Remember?
Yes, it's clear.
Okay. So now it takes me now, if you're all okay with the blood-brain barrier, it takes us to the last type of glial cells. Remember, all these are glial or supporting cells. So the last one we'll discuss is the ependymal cells. And these ependymal cells, they line the ventricles of the brain. I don't know whether you have covered the brain in anatomy. Have you? Can you let me know?
No.
You haven't done anything about the brain?
No, ma'am.
No.
Oh,
Not yet.
okay. So I had put a slide just in case you have not yet done. And I will first introduce you to what the ventricles are so that you know where they are and what their functions are. Now, in this diagram, I will ask you to focus on the diagram on my left. So actually, I can block this one for now so that we focus on this diagram. Now, within your brain, there are some hollow spaces. Actually, the other name for ventricle are hollow spaces. And that's why you have like the ventricles of the heart. These hollow spaces can be filled with fluid. So in the ventricles of the heart, you fill them with blood. Now, in the brain, the way the brain is structured, right in the middle, on your left side, we have one major hollow space, which is called a ventricle. Then on the right side, we have a second hollow space, which is also a ventricle. And these two spaces on your left and right, they form a C shape. Actually, it looks like your palm. Can you see? It's like you are holding something from your hand, and this is your thumb, this being the rest of your fingers, and you're trying to encompass something. So you have one on your left side and the other one on the right side. Now the two are going to have some extensions which come together and they coalesce and they form another hollow space in the middle. So we talk of the first and second ventricles, then we have the third ventricle here, which is a hollow space within the brain, and it links the first and the second together. Now this third ventricle will also form a narrow canal which will link it to another hollow space here, which is called the fourth ventricle. And these ones will form a continuous canal that goes right in the middle of the spinal cord. So if I were to cut a cross section here of the spinal cord, what I'm going to see is I have all this part which is cylindrical, shall we say, the spinal cord in this circular shape usually has a middle portion which is gray, and we said that the gray area has cell bodies and the non-myelinated neurons. And this other part is usually the white matter, which is very rich in myelinated neurons. Now, in every section of the spinal cord, we are going to find right in the middle there, it's called the central canal. And this central canal is filled with fluid, and it's the one that is extending from the fourth ventricle, and it goes all the way down to the tip of the spinal cord. And this central canal is the one that provides the nutrients that are required by the neural tissue. So where do we get these nutrients from? These will be synthesized by some special areas within the ventricles, known as the choroid plexus. We'll come to that later. But I wanted you to look at the structure of the ventricles and to know where they are. So if I go now to this diagram on my right, and I cut across, not really a cross section, you call it a coronal section. Can you see if I cut a coronal section through here, and then I look at it from the cut surface, I'm going to see the two hemispheres of the brain on each side. So this is one hemisphere and the other hemisphere. Now you can see right here in the middle, this dark area, this is one of the hollow spaces here. It's one ventricle. Then the other one is on the other side. You can see that they are separate, but they connect to a third ventricle, which goes down to the fourth ventricle. So those are the hollow spaces within the brain, and they are fluid filled. If I were to tilt this head so that I see the top of the head, and I cut a surgical section, remember this is corona, this is sagittal. So now what I'm going to see is part of one of the ventricles here. I've cut a section of one of the ventricles, and now I go to the third ventricle here, and this third ventricle goes to the fourth ventricle, which extends further down to form the central canal. Now in these areas there are some specialized area which we call the choroid plexus, and I think we find them mainly in the third and the fourth ventricle. These are the ones which allow material to come from the brain, and they select and they decide what goes into the ventricle. So they synthesize this fluid that is found within the ventricles and within the canal. And that fluid is what you call the cerebrospinal fluid. It's the one that nourishes the brain tissues. So now it also nourishes the spinal tissue. So all the cells of the spinal cord and the brain, they are nourished through this cerebrospinal fluid. So now we have already said, categorize the anatomy of ventricles, and you have said that you have that specialized area that synthesizes these, whatever the CSF. So let me go back to the previous diagram, which is discussing the ependymal cells. So the ependymal cells are the ones which line the ventricles. So the walls of the ones that are forming the ventricles, and of course the spinal canal, it is lined by these specialized cells which are called ependymal cells. And what they do is they separate the brain tissue from the space where we have all this fluid. So we don't want the fluid leaking out and filling all this space. So what it does, it selects what goes into the brain tissue, and you can see all this extension. That is how it will nourish the astrocytes, which will nourish the other cells. So the brain cells are all in contact, and they select what goes into these cells or to the neurons. Now, these ependymal cells within the choroid layer become specialized to form that choroid plexus, and the choroid plexus, its work is synthesis. They filter out, they take some of the stuff from the brain, sorry, it's on the other side. They take from the brain, and they are not from the brain, from the blood vessels. So they are enriched with blood vessels, so they'll take material from the blood vessels, pass through the specialized choroids plexus cells, and they release the material into the ventricles, and then from there it will be availed to the brain tissue. So all in all, to summarize, microglia are macrophage-like tiny cells, mobile, they clean up the brain space. Macroglia are made up of three types: the astrocytes, which form the blood-brain barrier, the oligodendrocytes, which myelinate neurons within the CNS, and the ependymal cells, which line the ventricles. Then we have glial cells of the PNS, as Schwann cells, and satellite cells. So if you don't want to remember all the details in that carried in that slide, you can use this diagram, which is showing you all those cells as I have described them. Within the PNS, we have satellite cells and Schwann cells. And then within the CNS, we have oligodendrocytes, microglia, astrocytes, and the ependymal cells. So, yes, Rutendo, you have a question?
Can you hear me?
Yes.
Okay, ma'am, can you please go back to the epidermal cells?
These
And
ones.
there's a pathway that you talked about from the conoid flexor cells as they move.
Choroid plexus cells do not move. They are just forming, they are part of the ependymal cells, but they've been modified. So most of these ependymal cells cannot synthesize cerebrospinal fluid. But the ones that can synthesize have been modified. So they are the cells of the choroid plexus. Is that okay?
Yes.
All right. So now from there we go to nerves, the excitable tissues, nerves, and the functional unit being the neuron. So in our previous course, 1331, we did mention excitable tissues, and we said parts of a neuron include: one, we have neurons will always have a nucleus. Then they usually have a cell body which may have cytoplasmic extensions like that. Now, one of these extensions may be modified to carry out impulses, to send signals. And when it comes to the terminal area, it may be spread to form what we call the terminal regions of the neuron. Within this terminal area, we can have what we call the the vesicles, and the vesicles carry neurotransmitter. So if we have the general structure of a neuron, which is the functional unit of the nerve, there are four main areas to look for. One, the receptor zone, the one that receives, takes information towards the cell body. That can be formed by the dendritic zone, where we have all these, they can change and they can take information that way. In a sensory neuron, we can also have nerve terminal, which can take information towards the cell body. But all in all, if these are the cell terminals, they can go and the cell body is located somewhere, and then they can take the information elsewhere. So this will be the structure of a sensory neuron with a nucleus and cell body between two axons. This is the structure of another type of axon, but another type of neuron. All I want to tell you is that you can have the receptor zone, the one that is receiving, can generate and receive information and take it in this direction, or you can have it in this format. Now, once the information goes towards the cell body, it will go to a place where action potentials are propagated. That is the second important part. Where do you propagate the action potential? In some places, we have a specialized zone, which we call the axon hillock. So you can have an axon hillock, and what you have in this axon hillock is numerous, very many voltage sensitive sodium channels. And when they are stimulated, they open and they allow a flood of sodium to flow in, and that flood of sodium changes the cell membrane to reach threshold and to initiate an action potential, which is then propagated. If you don't have an axon hillock, for example, if you have a sensory neuron here, now you find that the first area has a specialization which can allow it to initiate an action potential. And that area is what we call the initial segment. You can have the initial segment within the motor neurons, or you can have the initial segment within the sensory neurons. So if we do not have myelination, we still have that section that is capable of initiating an action potential, and this will happen in a non-myelinated neuron. Or you can have that initial segment that is in a sensory neuron that will initiate an action potential within a sensory neuron. So we have discussed two parts, the receptor zone, and now where you generate the action potential. Number three, we have the axonal processes. These axons are the ones which have the capability of transmitting impulses and passing them to the second cell. So these axonal processes will have either a myelin sheath or they will be naked. But either way, they'll have channels which can allow thresholds to be achieved and an action potential to be transmitted and to move along the cell or the nuclear membrane, the neuronal membrane. It will go all the way to the nerve endings, and the nerve endings comprise the fourth zone, which is where you release neurotransmitter. When you release this neurotransmitter, it can either go and land to a second neuron, therefore initiating an action potential in the second neuron, or the neurotransmitter may be targeting a muscle cell, and from there initiating an action potential along the muscle cell membrane, and therefore triggering an impulse within the muscle. So either way, the nerve endings are going to initiate action potentials in an adjacent cell. So 4 zones: receptor. We have the receptor zone. Number 2, the initiation zone for action potential. Number 3, the axonal processes. And number 4, the nerve endings. So I've already mentioned here and there the structure of the neuron. So we do have the receiving area or the receptor area. Mainly we have many cytoplasmic extensions which can easily be triggered to initiate an impulse. But you find that not really an impulse, to initiate a depolarization. And I'll tell you the difference. When you have a depolarization here, this depolarization cannot generate what we call an action potential, because the action potential, remember, it has a certain characteristic. We do not see that in the dendrites. When the dendrites are depolarized, they will allow a flood of ions to get in. Mainly, we talk of sodium ions. And depending on the amount that you have allowed in, they may hit the axon hillock. And when they reach the axon hillock, this is where they determine, do I reach threshold or do I not? If they do not reach threshold, that depolarization just diminishes, it disappears. But if they hit threshold, then they are enough to generate an action potential along the axon. Now, what I want you to do is on your own, I want you to draw the structures of the neuron, the functional unit which is the neuron. It will usually have a cell body with these dendritic ends, and it will have an axon which may or may not be myelinated. And at the end of the axons, they will branch to form the axon terminals, and each of the axon terminals will have a terminal valve. In this case, they are talking of a button. In some cases, you will talk of a knob. But what happens is that this terminal portion will bulge up, and it bulges because of the presence of the neurotransmitter vesicles. So, the parts you need to keep in mind are the dendrites, the cell body, the nucleus, the axon hillock. Nodes of Ranvier are those spaces or gaps between the Schwann cells. And it is very important to note about the nodes of Ranvier because these are the areas where depolarization occurs. Most of the depolarization, once initiated at the axon hillock, will jump to the node of Ranvier. So you hardly find any depolarization along here. And after it depolarizes here, this will jump to the next one. On the other hand, when you do not have a Schwann cell, then you'll find the depolarization will drag along the axon, just on the cell membrane of the axon. Now, if you remember, when I was describing the formation of the myelin sheath, I told you that you will have a Schwann cell like this with the nucleus. Usually the nucleus and the cytoplasm are towards the terminal part of the Schwann cell. But the Schwann cell will have wrapped around the axon several times, you can see. And as it wraps around, it squeezes out the cytoplasm. And the cytoplasm ends up just being on the peripheral part. So the middle part, the central portion that lays next to the axon, the yellow part is the axon. And you can see the axon has several microfilaments or microtubules which run parallel. So this axon, immediately surrounding it, will have that several layers of the myelin sheath. Of the Schwann cell. And between the two Schwann cells, we have a tiny gap which we call the node of Ranvier. So I gave an exam some time back, and I asked people to label these parts I'm putting with an arrow, I'm pointing at with an arrow. So I think those who had memorized this diagram, they gave me the wrong answer. What do you think this is? The blue point that I'm pointing at, what is it? Can you see the
Nucleus.
nucleus of who?
The Schwann cell.
Thank you. It is the nucleus of the Schwann cell. Now, I think somebody had memorized this point, and when they saw the Schwann cell, they told me this is the Schwann cell. So I need to make it clear. I think that's why I added this diagram, I added later, so that you can see the relationship between the nucleus and the Schwann cell, and then you see the nucleus of the neuron. So you have the whole of this is a Schwann cell, and each Schwann cell has a nucleus, and the whole of these is the myelin sheath. Okay. So I had told you that we'll discuss the myelin sheath in details, but I have already touched on it. Somebody has a question? Chikondi, mute yourself, please. Thank you. So I have already touched on some information here and there. Hold on. Just give me 1 minute. I direct somebody who is so lost. They're asking where the class is. So I hope either they have joined or they're in the second class, otherwise they'll have missed the whole lesson. Okay. So I've already mentioned that the axons can be myelinated, and they are made up of Schwann cells which wraps around the axon. And what makes up the Schwann cell or the myelin sheath? It's a lipid protein complex. Now, I'll take you back to the cell membrane. If you remember, cell membranes are usually made up of phospholipids. Many phospholipids. Then in between the phospholipids, we have proteins. These proteins can be transmembrane proteins, or they can be integral proteins, or they found on the periphery or on the inner side of the cell. So whatever the case, we will have a layer of phospholipids, and in between we do have proteins that can be crisscrossing, they can be peripheral to the outside, they can be more to the inside. So what happens is, remember that in this area I have cytoplasm, but I have phospholipids here together with the proteins. So when I oppose two layers of the membrane, so if I have, maybe I need a flat plate so I can work on. Just let me use this part. So if I have, this is the cell membrane, and it has two layers of phospholipids. Remember, the tails are facing one another. So I have cytoplasm here. So once I start wrapping around, I'll push the cytoplasm to one end, and what I will have is this double layer. Remember, this, you have squeezed out the cytoplasm, and what you'll end up having is one membrane, cell membrane opposed onto the other cell membrane. And now the cytoplasm is split in this area. Now this is the nucleus. So you can see as you squeeze out, if this is the axon here, as you roll it around the axon, you leave the two layers of cell membrane, but the cytoplasm is here. So if I wrap this around, what I will have is continuous layers of cytoplasm, of cell membrane, rolling around, just cell membrane, cell membrane, cell membrane. When you have that, what will be, if you had moved from the axon all the way to the peripheral of the Schwann cell, find that this thick layer is actually these phospholipid bilayers with some proteins. The question is, what makes up those layers? Is it just phospholipids, or do we have other components? And this is where we come to the composition of the the composition. It is made up of protein lipid complex, which rolls several times. Now, I have already said about its function when it comes to neurotransmission, and that not all cells are myelinated. And within the CNS, they are made up of oligodendrocytes. Within the Schwann cells, they are made up of, within the PNS, they are made up of Schwann cells. So all these points I've mentioned. Now, maybe what I can highlight are the neuropathies. It is very important that those myelinated cells stay and they remain myelinated. So in some cases, we have degenerative diseases whereby the myelin sheath is destroyed. It could be an autoimmune disease whereby you start attacking your myelin sheath and you destroy. Now, what happens is that it slows down neurotransmission, and with continued degeneration, you find that you lose muscular function. And a good example of this is the disease called multiple sclerosis. In multiple sclerosis, you find that the motor neurons that are supposed to be innervating your skeletal muscles, they start becoming demyelinated. And what happens is, with time, you start losing control of, even you slow down on activating your muscles. So you actually want to move your leg or your hands, you send the information, but they hardly move. It takes some time before they actually move. And with time, you even totally lose that control. Now, the problem is, if you do not stimulate muscles, they degenerate. So with time, if you have multiple sclerosis, even your muscles start to emaciate and you lose function. Now you find that some of these diseases, you can live with them over a period of time, but finally they end up being fatal. So this is one part that I needed to mention, which shows you the importance of myelination. Now, I want us to finish on the composition of the myelin sheath, so that I think I can start from there next time. It's almost time. So we say that we have both lipids and proteins. The problem is they are not uniformly distributed, whether it is within the CNS or those myelin sheaths of the CNS, they are not uniform. If you take the myelin sheath of the CNS and that of the PNS, again, there is a difference. So you find that whereas we have about 70% of the sheet being made up of lipids in the CNS, 80% forms the PNS. So if you take the Schwann cells, the cells or the lipid layer forms 80% and only about 20% is protein. If I go to the CNS, this drops down to about 70% and only about 30% will be protein. If I go to plasma membrane of most cells, you find that about most of it will be protein. 50% or even greater will be protein. So if you remember the function of those proteins, some of them are carrier proteins, some of them form channels, some of them are receptors, some of them are structural. So plasma membranes in these other general cells will have all those, because maybe their work is to transport material, their work is maybe to receive information, they have receptors. So all that will have a very high percentage of proteins. However, in the CNS, that function of the protein is diminished and the work is taken up by the supporting cells. For that reason, you don't need as many proteins, so the protein content drops. In the periphery, you also still drop it further because you have, in addition to the supporting cells, you also have a lot of connective tissues and blood supply. And therefore, those functions that would have been carried out by these other cells, we do not need the proteins. So in the PNS, most of the Schwann cells will have 80% lipids, in the CNS 70%. Now, the fact that they have high levels of lipids, they make the cells become very vulnerable to being attacked by toxins. Why? Because the toxins are usually hydrophobic and they'll go to that, they'll easily diffuse through the lipid bilayer. For that reason, I will give you a very classic example. When you are working in the lab, and you're using all these volatile fluids, hydrophobic fluids, you are told you must work in the fume hood. If you don't do that, it might cause, in the long run, it will cause brain damage. Why? Because you inhale those fumes. When you inhale, they will pass through your nostrils. And if you remember, we said that the olfactory bulb is very close to the brain. If you remember when we are doing some, when we are looking at sense organs in the last course, you find that the olfactory bulb, which is receiving the smell, it will take those fumes into the brain. And if you accumulate them, they'll go and sit in your myelin sheaths. With time, accumulated effect will start destroying your neurons. And that's why you're told you might have brain damage in the long run. So you must use a fume. Now, the other thing is, like when people take these alcohol, these adulterated alcohol, and they're taking methanol, and the methanol goes to the brain. It starts attacking some of the cells, and among the vulnerable cells are the ones of the retina. They cause retinodystrophy, those people become blind. So you find that you do not want those toxicants, those hydrophobic components going to your brain, for the simple reason they will attack your myelin sheaths. Now, the composition I have just mentioned, it differs whether you're in the peripheral or in the CNS. They will not have the same amount of lipids. In addition, if we take the lipids, the types of lipids, remember lipids are the glycerol plus the fatty acid chains. You can have a modification whereby you have these lipids are not just simply fatty acid chains, they may have additional of other components. So we have things like cholesterol, cerebroside. You can have an addition of a glucose unit forming like galactoceramide, sphingolipids. All these are different modifications of the lipids. This may appear in different amounts, so that in one part of the CNS, you may have more of sphingolipids, less of galactoceramide, and less of cerebroside. When you go to another part of the brain, then you have more of cholesterol and less of sphingolipids, more of this. So the amounts of each individual components may differ. But in all, you are going. Yes? Joel, you have something to say?
No, just continue.
Okay. Yeah, so I think with that, this was my last slide, because after that we'll go to the function, but I think it's already time, so I will not go to that. So I will quickly take attendance, and we meet at 1:20. That one will be a short one because it will just be explaining to you the theory behind the practical. So when you go to your practical session is on Monday, isn't it?
Yes.
Your practical session should be on Monday, and next week will be week 3. So we expect to start on the practical. So if I don't give you a free lab today, you won't have an idea of what we are doing on Monday. So quickly, let me take attendance. Nawal, are you there?
Present.
Sumaya?
Present.
Joel?
Present.
Atalina?
Present.
Emmanuel?
Present.
Angela?
Present.
Yusuf?
Present.
Kitonga? Sylvia? Not there. Gilo? Gilo is not there? Okay. Okay, please unmute yourself so that I can quickly take attendance. Kabanda Dorothy.
Present.
Kajiju.
Present.
Gloria. Yoko Perry. Kirwa.
Present.
That is Kirwa?
Yes.
Okay, Coril.
Present.
Natasha. Bukalanga. Victor. Victor not there. Ivy.
Present.
Brian.
Present.
Aisa.
Present.
Dennis Wanjiro.
Present.
Lutendo.
Present.
Lynette.
Present.
Lynette Mwangi. Lynette Mwangi.
Present.
Priska.
Present.
Sandra.
Present.
And Patience. Okay. Patience is not there.